Our first new grant recipient is Dr. Jim Dowling from the University of Pennsylvania. Dr. Dowling will receive $400,000 over the next 2 years for his project entitled Non-Viral Gene Therapy for Duchenne Muscular Dystrophy. This project explores innovative approaches aimed at addressing the genetic root of Duchenne. And before I turn it over, I'd like to encourage everyone to post their questions into the chat, as we'll open it up for questions after each speaker. Now, without further ado, Dr. Dowling, I'll turn it over to you.
Thanks, Lisa. I'm gonna share my screen. Can everyone hear me okay? Um,
Yep, we can hear you well.
Great. All right. So first, I want to say it's a great privilege and honor to be presenting today and also to receive a funding award from Defeat Duchenne. I have the privilege of having been involved with Defeat Duchenne for many years previously as a member of the Research Advisory Committee, and got to work with Lisa and several others on that. And also actually my interaction with Jesse's journey goes back to my time in Michigan when I first heard about it before I was a clinician at Sick Kids now 13, 14 years ago. So it seems a bit full circle to be coming now with a research project and really excited about the project and also very honored to have received a funded grant. So I want to just give a bit of the highlights of what the challenge that we're trying to address and what we're hoping to accomplish over this next 2 years. And I should mention this is a collaborative project between myself and Dr. Drew Weissman, who's a prominent clinician scientist researcher at the University of Pennsylvania. Hans, Dr. Lockmiller really introduced this topic, so I don't really have to go over this, but he highlighted the many different potential opportunities related to what I would consider the overall class of genetic medicines to treat Duchenne muscular dystrophy, and highlighted as well the challenges that these types of therapies are facing. So I think about these in two as being kind of two compartment therapies. There's the cargo, and that's the actual therapy that's being delivered, whether it's a small RNA like an antisense oligonucleotide, whether it's a gene itself in gene therapy, whether it's a gene editing reagent like Cas9 or one of the CRISPR reagents. And then there's the vehicle to deliver it. And in skeletal muscle diseases and Duchenne in particular, the big challenge has been that delivery vehicle. It turns out that it's very difficult to deliver medicines to all the skeletal muscles in the body. The current delivery strategies all have various shortcomings that really open up the question and challenge of whether new and different and effective delivery vehicles could be developed. And that's the goal that we're trying to address. So there is a delivery vehicle out there that is quite ideal in many ways, and that's called a lipid nanoparticle. And these are pretty ideal in terms of delivering the full range of genetic medicines. And so these are encapsulated lipids that have several different potential components in them. And they are a delivery vehicle that can very well encapsulate nucleic acids of many different sizes, everything down to the small antisense oligonucleotide that Dr. Lockman was talking about, to theoretically the entire dystrophin gene. The great things about them is that they are safe with very low toxicity. There's a lot of experience in humans of receiving lipid nanoparticles very safely. They're relatively inexpensive to manufacturing, which has been a challenge with some of the other genetic medicine strategies in AAV gene therapy in particular. And they're also very tunable. You can give them repeatedly. You can deliver some therapies that only need to be given once, but you can also have things that you can give multiple times and do that safely and effectively. So these are, again, all the different types of things that could be delivered with a lipid nanoparticle. So everything from those antisense oligonucleotides, which can be more effectively encapsulated and delivered in ASOs and given by themselves, all of the gene editing reagents, and then gene replacement therapy with full-length mRNA. So the big challenge with lipid nanoparticles is that they love to go to the liver when you're given IV, and unfortunately don't like to go to many other tissues. And in particular, don't go very well to the heart and the skeletal muscle, which is obviously the critical tissues for treatment for Duchenne. This has been the big barrier in the field of non-viral delivery for skeletal muscle disorders. In that whenever one gives these lipid nanoparticles to whether it's an animal model or to a human, they essentially go primarily to the liver. So this is the challenge that we're trying to overcome with our grant proposal. And our goal is to create really the first effective and safe muscle targeting lipid nanoparticles and to use those nanoparticles for the delivery of key genetic medicines for the treatment of Duchenne muscular dystrophy. And in particular, we are seeking to use this as a strategy for an alternative to gene therapy and to use it as a way to deliver the full length dystrophin mRNA as a gene replacement strategy. So this is how we're going to do it. This is a very busy slide, and I apologize for that. But we're doing this in two different aims. The first aim is to develop our muscle targeting lipid nanoparticles, and we're doing that through a very comprehensive and systematic process. Where first we're creating a library of different lipid nanoparticles, and the library will differ based on the composition of the lipids, but most importantly, by the presence of targeting antibodies. And we've selected a library of antibodies that we will recognize things that are on the skeletal muscle already, and basically serve as honing devices for the lipid nanoparticle. So basically adding a signal onto the nanoparticle that will then match a signal that's present on muscle as a way of drawing the nanoparticles to the muscle. We're also trying to innovative strategies to detarget the liver. One is to remove the parts of the nanoparticles or the receptors on the liver that contribute to nanoparticle uptake in the liver. And the other is to try to enhance the delivery of nanoparticles to muscle by creating increased blood flow into the muscle beds. Once we've developed these library of nanoparticles, we're going to test them systematically in a mouse model. And this is work that has to be done in an animal model because doing them just solely in cells does not recapitulate the challenges of the barriers to the delivery. So it's very easy to actually get nanoparticles into muscle in a dish because you've removed the things that prevent nanoparticles from going to muscle in a whole body. And so we're doing all of our screening in the mouse as a model system. And we're first just screening for the best nanoparticles to go to muscle. And then we'll do a secondary screen using a clever marker mouse that enables us not only to tell whether or not we're delivering, but also enables us to see whether we're delivering to the stem cells and the satellite cells, and also to which percentage of the muscle nuclei that we're delivering to. And in this way, we're able to develop the best muscle targeting lipid nanoparticle. And once we have that, we will then go on to test for safety and effectiveness, mRNA-based gene replacement strategy for Duchenne, encapsulating the full-length dystrophin mRNA into our best lipid nanoparticle, and then delivering it to the MDX2V mouse model of Duchenne muscular dystrophy. And this is basically the paradigm in which we will do that. So as I mentioned, this is something that has not yet to be really successfully done. And so why do we think we can do this when others have maybe not succeeded in that space? And that primarily has to do with the very strong track record that we have, and in particular that Drew Weissman's group has in developing targeted nanoparticles. So he is now his group has developed lipid nanoparticles that target several different hard to target tissues, including the endothelium, the immune T cells, and also to several stem cell compartments. And so the main strategy that he's employed and that we will employ in this proposal is to add these tags onto the lipid nanoparticles. In this case, showing an antibody tag that enables that honing of the nanoparticle to the tissues of interest. And interestingly, adding these tags also by themselves somehow detargets the liver. So whenever you put a tag on a nanoparticle, it tends to make less of it go to the liver. So you accomplish two things: directed targeting to your tissue of interest and detargeting of the liver. A recent example of how the Weissman group has done this is by adding antibody-tagged lipid nanoparticles that hone in on these immune cells called T cells. And he's used these to deliver specific receptors to turn T cells into CAR T cells as a potential anti-cancer therapy. Right now, CAR T cells are used on an individual basis, but they require the removal of the blood cells from the body, then the targeting, and then the adding back. And this strategy to be done in an individual without actually having their T cells removed. Actually, there is some applications of the specific strategy related to fibroblasts and fibrosis. That is a secondary consideration with nanoparticles for treatment of Duchenne, but is outside of the scope of the current proposal. This is just showing the effectiveness of these in vivo CAR T cells that are done through the targeted lipid nanoparticles and how they can be used to deplete a specific cell population in the spleen. Probably more relevant to our proposal at hand is the fact that the Weissman group has developed heart-targeting lipid nanoparticles. And this gives us a lot of encouragement that we can similarly develop skeletal muscle targeting lipid nanoparticles. And at the very least, we can build off of these nanoparticles as a way to start treating the cardiomyopathy of DMD. In this study, showed both the effectiveness of adding a specific type of lipid, a different kind of lipid within the nanoparticle to make ones that go to the heart over a standard formulation, and then also using this lipid detargeting strategy of reducing the amount of ApoE to detarget the liver. And that's just an example of how it was used to effectively deliver an siRNA, so a small RNA therapeutic, to the heart to produce gene knockdown. So you could imagine a similar approach could be used to deliver an antisense oligonucleotide to promote exon skipping for DMD, or again, to deliver the full-length dystrophin mRNA. So to summarize, we have previously demonstrated the ability to target nanoparticles in hard-to-reach non-liver organs and tissues. We have been able to deliver a range of genetic medicines using these nanoparticles, including ASOs, CRISPR reagents, and mRNA. And we've shown that the combination of antibody targeting with this ApoE knockdown both detargets the liver, but also enhances targeting to tissues of interest. So we propose to use the same strategy to achieve muscle targeting. And just to give a little teaser, and this has been in collaboration with and through a previous grant that we had with the National Research Council in Canada, we have developed some nanoparticles that are showing some muscle targeting. They're not optimized, and we have a lot of work to go. But we have been able to show the ability to deliver not DMD, but a smaller transgene, the MTM1 gene, which is a disease-relevant gene, using nanoparticles after IV injection to go to the muscle. And that we've been able to show that these can ameliorate parts of the phenotype of the mouse model of X-linked myotubular myopathy, which is caused by loss of the MTM1 gene. So we can add back MTM1, and that can produce some benefit. So this gives us the first hint that our strategy of tagging lipid nanoparticles can overcome the barrier of muscle delivery and develop muscle trophic nanoparticles. So to summarize, there's in my opinion, a critical unmet need to develop new delivery vehicles for the treatment of Duchenne. Lipid nanoparticles offer great promise, but historically have not been able to be targeted to skeletal muscle. Based on previous success in our preliminary data, we believe that we will be able to develop very potent and safe muscle targeting lipid nanoparticles that will then open up the entire universe of genetic medicines for the treatment of Duchenne. And with that, defeat Duchenne for supporting this project, and we really look forward to embarking on this endeavor and within our 2-year period delivering lipid nanoparticles that can treat DMD.
Thank you, Dr. Dowling. That was great. Can your potential treatment work with exon duplication?
So the short answer is yes. So if we successfully develop muscle targeting lipid nanoparticles, there's a very broad range of types of therapies that could be encompassed within them. So some of the strategies for treating duplications, including using CRISPR-Cas9 editing to cut out the duplications. There's also some use of alternative start sites with CRISPR activation. Any of those reagents, the gene editing reagents, fit very well in the nanoparticles, and it overcomes the problem that the viral vectors have, which is that the editors are often too big for the AAV gene therapy. So in principle, yes. And, while we're focusing on the mRNA therapy approach, because it would be mutation agnostic in terms of treating DMD, we plan to make the nanoparticles available broadly so that individuals working on other strategies can take advantage of this as well. So yeah, I guess a long way to say shortly, yes, the duplication strategies could be treated with LNPs.